My name is John Moore. I am a new Associate Dean for Faculty Affairs in the College of Arts and Sciences. And it is my pleasure to introduce Dr. Nicole Riddle for the Haddin Forum's inaugural session of the 2021-2022 academic year. Dr. Riddle is an Associate Professor of Biology at UAB, where she teaches courses in genetics and epigenetics, runs a research lab, and works as an investigator for such groups as the UAB Nutrition Obesity Research Center. She obtained her PhD in evolutionary and population biology from Washington University in St. Louis, where she worked as an assistant research professor and visiting scientist until her arrival at UAB in 2012. Dr. Riddle studies epigenetics, which she describes as, quote, a research field that focuses on heritable changes in phenotypes that are not associated with changes in the DNA, end quote. She has several national current and completed grants from such entities as the National Science Foundation, and has won numerous awards and fellowships, such as the John R. Durant Award for Excellence in Cancer Research here at UAB, as well as a Commission on the Status of Women Award for her efforts as a female leader in the STEM disciplines. Dr. Riddle's peer-reviewed publications number close to 50, and she has spoken across the country as an invited lecturer and conference presenter. Today, Dr. Riddle will speak to us on genetic pathways controlling exercise response, a topic that will be of great interest to many of us vis-a-vis our body's various reactions to exercise. Her lab analyzes a classical genetics model system, the fruit fly, Drosophila melangoster, to understand variation in exercise response. Dr. Riddle will present to us on the ways in which, quote, sex and genotype have a strong impact on exercise response, and that the genetic networks controlling exercise response depend both on sex and the response variable, end quote. These findings suggest that exercise regimens might be custom tailored to individuals based on sex and genotype in order to maximize the efficacy of an exercise treatment. We will take your questions for Dr. Riddle at the end of her talk. Feel free to submit them into the chat feature of Zoom or to save them for the end. In the meantime, please join me in welcoming Dr. Nicole Riddle.
Thanks very much for that really kind introduction. I appreciate the opportunity to be here today and tell you about some of the work that my lab is doing. As Dr. Moore said, I'm going to tell you today about how we use the fruit fly Drosophila melanogaster to study exercise and exercise response. So I'm going to tell you about the tools we developed so we can actually exercise flies and some of the findings that we have learned regarding exercise responses and the genetic pathways that control them. Okay, so just to give you a little bit of background, as most of you are probably familiar with, obesity is a big problem across the world. Obesity is a risk factor for a variety of common diseases, including diabetes and cardiovascular disease, which you probably knew, but it's also a risk factor for other diseases, including cancer and infectious diseases. It can lower life expectancy, and in many cases, it can lower quality of life. And it also increases healthcare costs for individuals and societies. In the US, for example, over the last couple decades, well, I guess it's more than a couple at this point, over the last several decades, the obesity rates have dramatically increased. And so if we look at this chart here, we can see that in 2010, according to the CDC, we had the vast majority, 70% of the population, be either overweight or obese. And that's quite a bit. And so that's quite of a big problem. If we look a little closer to home and look at how the obesity rates look like, if we look at them by state, this is data again from the CDC for 2020. And we can see that there's very few states left that are in yellow or in green, where the obesity rate is below 30%. And there's a lot of states where the obesity rate now is exceeding 35%. If we look at Alabama, the obesity rates here are 39%. So 39% of our population here is obese and has to deal with the health effects that come along with that. The rates differ between the different racial groups in our state. So for example, for Black Alabamians, the obesity rate is 46%. For Hispanics, it's 35%. And for white Alabamians, it's 34%. So all across the population in our state, we basically are dealing with this health problem. So as most of you also probably know, to combat obesity, and in theory, that's very simple, right? Because you need to shift the energy balance. You basically need to somehow either decrease the energy intake or increase the energy expenditures of a person. So either we can modify diet or we can modify the activity levels and the energy expenditures. And that basically means exercise. And often the recommendation really is to do both, right? If you heard the health professionals talk about this, we really should be watching both of those things. But basically it looks like exercise might have an important role to play to help us deal with this, what's called obesity epidemic. Now, the benefits of exercise actually go way beyond helping with weight control. So exercise strengthens bone and muscles, and it helps reduce a variety of risk for diseases. So we have, it reduces the really common ones, diabetes and cardiovascular disease, but it also helps with, cancer prevention, and it can actually really prevent premature death. And the really nice thing about exercise is that for the type of exercise that most of us do, which is really different from the elite athletes, for example, exercise has very little risk. It's actually not very risky to do. And despite all of this benefits and these low inherent risks, only about 50% of Americans actually meet the recommended guidelines for exercise. And you might want to think about why that is, right? And maybe you're one of these 50%. I mean, half of us, I guess, obviously meet that. Or maybe you're among the 50% that don't meet that. If you're among the 50% that don't meet these guidelines, you might have had some of these experiences where you've started an exercise regime and you struggled to hang on with it and you toughed it out, and then you didn't see the result that you wanted. And that's a really common experience for people. And this is the question that my lab is started out with. We were really interested in this feature of variation between individuals. So exercise isn't really different from many other traits that we have and that we can measure in a human population or any other population, where there's lots of variation between individuals. And that's shown in this diagram here for body shapes in humans. There's lots of variation if we just look at the people out on the street or in our lecture rooms, right? And there's variation for many, many traits. And exercise and how we respond to exercise is also one of these traits where we see lots of variation. So when we think about exercise and exercise responses, there are lots and lots of things that we can measure, right? So individuals pursue exercise for a variety of reasons, right? So some individuals might want to lose weight, other individuals want to build up strength or endurance, or they would like to improve their muscle tone. There's people that would like to maybe lower their cholesterol or get their triglyceride levels under control, or improve their insulin sensitivity, right? So these are all what we call response measures that we can look at after an exercise treatment and see how they were impacted by the exercise. What all these response measures have in common is there's immense variability in this exercise response. So for all of these exercise response measures, we will see variation between individuals. So if we put whole bunch of people on the same exercise treatment, we will see people that have no response, people that have the expected and wanted response, And then we sometimes even have these people that get the opposite response. So the opposite happens of what we really wanted to happen. And so that's basically what we're trying to understand. Where does this variation come from? And we're focusing on the genetic component of this exercise response. Now, why might we think that there is a genetic component to exercise response? And there's a couple of data points that support this idea and that make it worth investigating. So one of the, I guess, oldest data points comes from the observation that when we have different strains of lab animals, and the older data comes mostly from rodents and mice, we can see that different strains of mice, so different genotypes, have different running behaviors. So there's animals that will, if you give them a running wheel and put it in their cage, they're super excited. They're going to be on there for hours every day, and they're just going to run because it's fun and that's what they want to do. Then there's other mice that will go and see that running wheel and they're like, oh yeah, nice decoration, but they're not going to actually go use it because they prefer not to. Something that's maybe more familiar to you is dogs, right? Different dog breeds are genetically different. And there are some that need to be walked, 3 times a day. They can't get enough of walking and they need to run around. And then there's other kinds of dogs that are much happier not walking all that much. And that's what they prefer. So this tells us that there is a really good likelihood that the preferred level of activity, so that your preferred amount of exercise, and also the responses to it are likely genetically influenced. We also know from work in things like fruit flies and mice and worms that there's mutations that will impact movement behaviors. And so we can find mutations that will make animals hyperactive. So they will actually be way more active than normal. And then we can find mutations that make the animals hypoactive. So they will show less movement behavior than normal. And then there is a little bit, very little amount of data that suggests that there are certain genetic variants that are overrepresented in elite athletes. So if you look at the genome of Olympians, for example, you can find that there's certain alleles at specific loci that are showing up in these athletes way more often than in the general population. And so all these things tell us that there is most likely a genetic component to how we all respond to an exercise treatment. All right, so with that, let me introduce you to the study organism that we use. So we use the fruit fly Drosophila melangoster that you might be familiar with from your kitchen. If you have some overripe fruit, they tend to show up and be a pest. But they're actually a very, very nice model organism for us to use. They're very small. They've been in the lab for about 100 years and been used by geneticists. They have a very fast life cycle, so we can go within 10 days from one generation adults to the next generation of adults. The adult female will lay eggs, the embryo will develop for about a day, and then a larvae will hatch. The larvae goes through 3 growth stages, and then it pupates, and after the end of 10 days, we have a new generation of flies, which is very nice and fast. Because they've been in the lab for so long, we have lots of genetic tools available for use with them, so we can manipulate their genome in lots of different ways. We also have a really high quality genome assembly. And from this genome assembly, we know that about 75% of human disease genes are also found in our fruit flies. And so we know that many of the results that we will find in the animals will translate to people because most of the metabolic pathways are really well conserved. And so that is why we use the fruit flies. You might want to ask yourself, also why we don't actually want to study people directly, because that would be on some level more convenient. But from a geneticist standpoint, humans are a terrible system to work with. Right? We're interested in the genetic pathways that control exercise response. And that means we would like to keep everything else the same for these animals, right? And people really don't tend to like it when you tell them, well, I would like you to exercise from 8 to 10 in the morning. And then I would like you to just kind of sit around and do nothing in this very temperature controlled place where nothing special is going on. We're going to give you the same food all day long for about 2 weeks or so. And then, these are not experiments that you can nicely do. As a geneticist, we also would like to, obviously get specific genotypes so we can test things. And that's, again, something we really can't do with humans because that's just not feasible. And so if we want to have a really nicely controlled experiment where we really only look at the genetics, where we have lots of replicates, so we have hundreds of genetically identical individuals, flies or other model system like flies are really the way to go. All right. So you might ask yourself, how do you actually exercise a fly? And it's not a little treadmill. Instead, what we do is we exploit this behavior that the flies have that's called negative geotaxis. What this means is that when we have the animals in a small enclosure, these are usually little cylinders or bottles. In the old days, they used the old glass milk bottles that you could have. But in any enclosure that you have the animals, they like to hang out at the top. So if you tap them down in their little enclosure to the bottom, the first thing they do is run right back up to the top. And obviously that's some way that we can basically manipulate to get them to exercise. The very first fly exercise machine was developed by the Wessels lab and published in 2009. And you can see it right here. It's basically a platform where the enclosures of the animals are standing on, and there's a lever that lifts them up and then drops them multiple times a minute. This works really well because as soon as it drops the platform, the animals fall to the bottom of the enclosure and then start running up. But what they've also noticed is that this is pretty rough on the animals to lift them up and drop them over and over an extended period of time. And so when we started this work, we were looking for an alternative. And we worked together with Dr. Laura Reed's lab over at UA to develop and validate this alternative approach to fly exercise, which we call the Treadwheel. And what it basically is, it's a rotisserie for fly enclosures, if you can imagine that. So we have a rotating rod that you can see right there. And there's clamps where we attach the fly enclosures. And it basically rotates the enclosures end on end. And so while we're rotating this, if we're rotating this slow enough, the direction of up changes and the animals just try to catch up with where up is. And so they keep running. So here's the schematic of the machine, and here is what it looks like in real life. In real life, we have 4 different rods, each has 12 clamps on there, and so we can exercise 48 sets of flies.
Which outside chopped. I need a large order of fries. And then I need the barbecue chicken sandwich. Can I get that chopped?
Let's see. There we go. That should fix it. All right, so here's our exercise machine. The one thing that we still needed after having this exercise machine was we wanted to have a way to actually measure how much the animals were exercising. And so for this, we modified a machine that was already commercially available for fly geneticists, and that was a Drosophila activity monitor. This is a very fancy machine where it has the fly enclosures in this grid, and each fly enclosure has a set of laser beams that bisect the middle of the fly enclosure, which you can see here on the right. If a fly crosses that midline of the vial, the laser beam gets disrupted, and that creates a little electrical signal, and it can be counted. So we can count every time a fly goes through the middle of that vial. And that basically gives us our measurement of how much the animals are moving. And so then my student Lewis, who is responsible for the vast majority of this work I'm talking to you today, he generated the rotating exercise quantification system, or REX, which basically meant he took this activity monitor and he put it on a rotating arm. So now we can measure the activity of the animals without rotation by just not turning the machine on, or we can start rotating it just like on the treadwheel. We use the same vials for the same machine, both machines. And so we can measure how much they're actually exercising when they're rotating. Okay, so before I'm going to show you actual data, I want to introduce you to our study population. Our study population is the Drosophila Genetics Reference Panel. And this is a strain collection of 200 fully sequenced inbred fly lines. They were derived from females that were collected in the wild. And the wild here is the farmer's market in Raleigh, North Carolina, where the McKay Lab in 2012 grabbed these animals and then started breeding them from there. They're extremely well characterized. So these have been used in, I would say, probably over 100 studies by now. And so we have information about these fly lines, about their olfactory sensation. We have information about their circadian behavior. We have information about their level of aggressiveness and all kinds of other things, basically anything you might want to think of. And then we have an automated analysis pipeline that if we measure new phenotypes, we can integrate it with all this genomic data and other phenotypic data that's available for these lines already. And so with this set of animals, we started our experiments. And so here is the experimental setup for the very first experiment that we did. We collected our animals under very strict environmental conditions. So they get 12 hours of light dark, 70% humidity. They're all on the same food. There's always the same number of animals in each enclosure. We grow them, we collect virgins, we age them until they're 3 to 7 days old. And then we measure 10 vials of 10 animals for each sex and genotype combination. We put them in the Rex machine and we measure for 1 hour without any rotation. And then we turn the machine on and measure again, how much do they move with rotation. And here I'm showing you data from just 4 different lines. So if we just focus on the left first, that's where we have the data without rotation. Each of the colors is a different genotype, and the first box is the male, the second box is the female. On the y-axis, we have the activity level that we measure. So how often did they cross that beam in a specific amount of time? And so what you can see here, just looking at the left and without any rotation, we can see already that there's differences between these genotypes. So these 4 different strains of flies have different levels of activity, even if we just leave them alone and do nothing to them. And that's visible if you compare the yellow guys, for example, to the green guys that have much higher activity levels. You can also see, especially in that green strain right there, that the males and females don't do the same thing. The males and females actually behave very differently. Now, if we go over and look at the data on the right, which has the data from with the rotation where we're exercising them, the first thing you can hopefully notice is that all the measurements shift up. So all of the measurements shift up, which means the animals are actually working more. They're running around more with the rotation than they do without. So they're getting some exercise. How much more exercise, again, they're getting really depends on the genotype, though. Some of them increase by a lot, and some of them increase only by a little. And again, the males and females differ really in the way they respond to this exercise treatment. So the rotation is successful in inducing exercise, but it does so in a genotype specific way. So each of these 4 different strains behaves differently, and the 2 sexes behave differently. So the second basic experiment we did with our new machine was to look at 2 different exercise treatments. And what we compare here on the left, we have a 2-hour rotation, just straight rotation. We just turn it on, leave it on for 2 hours, and then turn it back off and measure throughout the 2 hours. The time is on the x-axis, how much the animals move. Again, what you can see is that there is a really big difference between these animals, between the different strains. The green strain obviously is way more active than the other ones, but they all move around quite a bit over that 2-hour period. On the right is a different exercise regime where we did what we call interval training with them, where we turned the machine on for 30 minutes, gave them a 10-minute break, turn it on for another 20 minutes, gave them another 10-minute break, and then turn it on for a last 10 minutes. And what you can see here clearly is that the animals increase their activity levels, especially if you just focus on the green one, which is just so extreme. They really start running around like crazy when you turn on the machine. And then they stop when you turn the machine back off, right? So that they really use that break. They just settle back down, take a break, and then when you turn them back on, they start running again. This is the same for all of the 3 lines that we have on here, but the green genotype is very much more extreme. But this really tells us that we can actually test different exercise regimes with this machine, that we can try different types of exercise patterns that people might want to use. For the rest of the talk, we're only using the 2-hour straight exercise block because that ended up being the simplest thing to implement and to basically handle for us for a starting point. So with that, we moved on to like looking at all those lines in our strain collection, those 200 different lines. And what I'm showing you here is the data that we collected without the rotation first. And this is the data from females. I'm going to show you quite a few of these graphs. So on the x-axis, you have all the genotypes. So each one of these little bars is a single genotype. And then on the y-axis, we have the average activity measured as these beam crossings. And what we can see here is that these females, there's some of them that basically just don't move at all when we don't disturb them. And then there's some of them that have about the 50-fold increase in activity levels. So we have lots of variation in activity levels among these female flies. Now, I'm adding on the right here for you what happens when we turn the machine on. So again, on the x-axis, we have all the different genotypes. On the y-axis, we have the activity. And I think what you can see pretty clearly comparing the left to the right is that everything shifts up, right? So we're clearly increasing the activity of these animals by rotating them. So they move more now. But what we also still have is we still have animals that basically don't move at all here at the bottom left of the graph. And then we have animals that have about 150-fold increase over these guys at the bottom in their activity levels. So there's a wide range of activity levels in these strains of Drosophila. So lots of different things happening. All right, and so on this last graph, I'm adding the males on the bottom for you. So again, on the to remind you, on the left, we have the baseline without rotation. On the right, we have with rotation. And at the bottom now, we have the males. If we're comparing at baseline, so we're comparing A to Z, we can see that the males move a lot more than the females do, just when we're leaving them alone at baseline. So males are much more active as a group than the females are. And if we compare C to D, the males also increase in their activity levels with the rotation, but much less so than the females. So what this data here shows us is that this strain collection has lots of variability in both the baseline and in the exercise activity levels. So there's lots of variation, and variation means we can go look at the genetics. So let me just summarize what we have here, what we've looked at so far. So I've showed you that we have a machine called the Rex that allows us to measure the activity levels of the exercising flies. We also can show clearly that activity levels increase when we rotate the animals. How much activity there is in the animals at baseline depends on the sex and genotype. And that's also true with the rotation. And this variation really depends on that there's basically lots of variation with or without rotation. And that means we can use these animals to try to get at the genetics. Okay, so that's basically what the next set of slides is. So variation between individuals has lots of different sources. Environment, you can think of diet, where those animals live, what they eat, what else they do other than exercising. But we try to keep all of that the same. That's why the fruit flies are so nice, because we have them in their nice little bottles and nothing much changes. And so we can really focus on just the genetics because we try to keep everything else the same. The tool that geneticists often use to try to get at the source of a variation is called a genome-wide association study, or GWAS. There's a lot of fancy math involved here, but the principle is actually relatively simple. So we basically look at lots of different places in the genome, and we're trying to understand if the genetic variation is associated in some way with the trait that we're looking at. And so if you look at our diagram here, you're already familiar with that top part of the diagram where you have the different genotypes on the x-axis, and we have their phenotype and the activity level on the y-axis. Underneath there, I have given you basically an example of a genetic locus where there's two alleles. There's a blue allele and there's a green allele there. If we were to average out all the lines that had blue and calculate an average, and then calculate an average for all the lines that have the green allele, there would basically be no difference, right? So the blue and the green allele are pretty much randomly distributed amongst our phenotype, right? There isn't any clustering at the bottom or at the top for green versus blue. So there's no association between the genetic variation at this locus and the phenotype. Now, in the second example here, we have a third allele and it's red. And the majority of the red allele is found at the very bottom of the distribution. And so now if we would go in there and calculate the average phenotype of the red ones and compare it to the average phenotype of the blue or green ones, that would be different. And so now we have what's called an association between that spot in the genome and our phenotype. And that means this is a spot in the genome where we're probably going to want to go and look, what is that gene? And is that possibly really linked to our phenotype? Does that gene make our animals be less active? So that's what we would want to look at. And so this kind of a calculation, looking at the distribution of the alleles in relationship to their phenotype, we do for thousands and thousands of spots in the genome. The data that you get from this kind of analysis is usually plotted in these kind of graphs, which are called Manhattan plots, because I guess it kind of looked like their skyline to somebody. The chromosomes are plotted on the x-axis, and every one of these little spots is a specific spot in the genome that we looked for this kind of an association between the genotype and that phenotype. And every time a little dot is above the significance line here at 5, it tells us that this is a significantly associated locus that we should look at more. So these are the potential candidate genes that control animal activity. And the graph on the left is for the baseline activity, and the graph on the right is for the exercise activity. What you're supposed to take away from this basically is that there's dozens of loci, so dozens of space spots in your genome that contribute to the variation in activity levels in these animals. So there's lots and lots of different genes that contribute to how much an animal will move. So the next thing we did was we obviously wanted to know what these genes are, right? And so since we know where all these spots are, that's where all those nice community resources and the nicely annotated genomes comes in helpful. We can now go into the genome and look at what these genes are. And all the genes in the genome in most of our model organisms have been annotated with specific terms that tell you about what these genes do and what organelles they function in and what tissues they function in. And so we can basically then go in and do what's called GO analysis or gene ontology analysis that will tell us what types of genes these are and where they're functioning. And here in this graph, you have I've given you a selection of the genes that are significantly enriched. So we basically find more genes of this type among the group of candidate genes that we've identified in our GWAS than we would expect by chance. And so the little gray bars are the ones that are observed, and the black black ones are what we would expect if it was just chance. And so you can see all of these ones here receive way more in our grouping than we would expect. And what's interesting here is that a lot of these have to do with the central nervous system. So there is the axonal growth cone, there's neuromuscular junctions, neuron recognition, so axon guidance, and then also stuff like behavior. So this tells us that genes linked to the central nervous system are actually very important for determining how active an animal is. And that's really not surprising. It's actually kind of nice that it worked out that way, because you would kind of expect that. So this part of our study basically showed us that, we have lots of variability in activity levels, and we can then use this this variability to identify over 300 genetic variants that are responsible for setting activity levels. Interestingly, the genetic variants differ between the two sexes. So what's important for setting an activity level in males and females is not necessarily the same. So there's different genetic networks that work in males and females. And then when we look at the functional categories of these genes, we find a variety of them, but many of them are linked to the central nervous system. All right. So this was basically a nice proof of principle that we can use this system to try to understand exercise and how much animals move and how much they exercise. Next, we really wanted to get to those exercise responses, right? We wanted to actually understand how different the animals are in their responses to exercise. And so for this, we use a very similar experimental setup. We again grow our animals under really controlled conditions. We have 10 vials of 10 animals per sex and genotype, but now we have an exercise group and a control group. The exercise group gets onto the exercise machine for 2 hours per day for 5 days, while the control group just gets to sit in the incubator. So we handle them exactly the same, but they don't get onto that rotation machine. After the 5 days are over, we measure their climbing speed. So we measure how fast they are at climbing from the bottom of the vial to the top, because we were thinking that with all that running, maybe they got better at climbing up the side of the vial. And we also measured their weight, because we wanted to understand if their weight might have changed with all this exercise that they were doing. And so here is the data that we collected for weight. And let me just get you oriented on this graph. So we have, again, on the x-axis, we have all the different genotypes, so all the different strains. On the y-axis, we have the weight of the flies. On the left, we have the females. On the right, we have the males. And on the top are the controls, and on the bottom are the ones that exercised. So if we just look at the controls first, we can see that there's a lot of variability again between these lines, right? So there's lots of variation in how much a fly will weigh depending on its genotype. We can also see that the females tend to weigh more than the males. So females are usually bigger, so they weigh more. And we can see that very consistently. If we look at the treatment animals, the ones that exercise, we can see the same thing. There's a lot of variability. Females are heavier than the males. But if we compare the top to the bottom, there is not an obvious difference. Not like when we compared the baseline activity to the rotation of activity where everything nicely shifted. Here, we can't actually see a general shift that's happening. So the weight is very strongly influenced by sex and genotype. But just looking at all of them overall, we can't actually see a strong impact of the 5 days of exercise treatment that we had these animals do. So to see the impact of the exercise treatment a little better, we calculated the change in weight by subtracting the control animals from the exercised animals. And so this is shown here for females on the left and males on the right. And what you can see here is that there's all kinds of stuff that happened to the animals in terms of their weight, right? So we have some animals on the left side of this graph that gained weight with exercise. We have other ones of the right side of this graph that lost weight with exercise. And then we have some in the middle where basically nothing happened. They exercised, but not a whole lot was going on. And that's the same for the males and females. So basically what this tells us is that how your weight responds to an exercise treatment depends, first of all, on your sex. It depends on your genotype, and you can basically get any outcome of this kind of an exercise treatment. If we do the same kind of analysis for climbing speed, we get very similar answers. So again, we have the females on the left part of this graph, we have the males on the right part of the graph. The top is our control animals, bottom is our exercised animals, and instead of weight, we're now measuring climbing speed, basically. We can see that there's variation for all of these, right? So we have variation among these lines again. And here, the males are better at climbing than the females. And that's consistent between our controls and our treatment animals. But if we compare the top to the bottom, just like with weight, there isn't really a strong shift that's induced by this 5-day exercise treatment. So you don't really get, there is not a general trend for everybody to get better at climbing once you go through this exercise treatment. But there's definitely a huge impact of sex and genotype on how well you can climb up the side of a while as a fly. We can then do the same analysis and just look at that change that happens. What is the change that we are generating by the exercise treatment? And just like with weight, we have animals that basically get better at climbing. We have animals that get worse at climbing, and we have animals that really don't see a change in their climbing ability at this point. So exercise responses, just looking at weight or climbing speed, are very similar to what we see in humans, in that we can see all kinds of responses, and there is not a simple predictor at this point that lets us know how an animal will respond. Given that we're working with this nice population that is really optimized for running these genome-wide association studies, we then try to run an association study to look at what are the genes that are given us these changes in weight and what's important for that. And this is a slightly different graph here. The lighter the color of the dot, the more significant the change was, and the bigger the dot, the more enrichment did we see. So the more genes of this class did we detect among our candidate genes. And again, here we see a variety of categories of genes, but again, a lot of them have to do with the central nervous system. So we have synapse organization, regulation of axon guidance, and regulation of axon extension, for example. So we have central nervous system genes again, but we're also detecting a new class of genes, and they have to do with signaling. So signaling between tissues and signaling between cells that we were pretty excited about. So with that, to summarize this part of the talk, we basically have fly strains that vary in their response to exercise, both in terms of weight and climbing speed. Like in humans, we see that there is positive responses, negative responses, or a complete absence of responses. The sex and the genotype are incredibly important for how an animal will respond to the exercise. And we find an interesting class of central nervous system genes again that seem to be controlling how animals respond to exercise. Okay, so there is a few more data slides that I wanted to share. And they have to do with us trying to understand how do these activity levels and these changes, these exercise responses relate to each other. So one of the first things we wanted to look at was how these activity levels of the animals correlate with lifespan. One of the things that's a very common perception is that if you're moving more, if you're more active, that you are a healthier animal and that you should be living longer. And so here we're correlating the activity levels on the x-axis with lifespan on the y-axis. And if that's really true, if that a more active animal would live longer, what we expect is that the dots here would fall on a diagonal that's going from the bottom left to the top right, right? So we should see some kind of positive slope. But instead, we really basically see nothing. There is not a significant slope here, and there's not a significant relationship between activity levels and lifespan. And that's true for the basal activity without the rotation, but also for the activity that they show with the rotation. So interesting, in these animals, there is not an obvious link between how much they move and their lifespan. Another thing we wanted to know is if the amount of additional activity that we're inducing by the rotation, so how much does the activity increase with the rotation, that this would be predictive of how well the animals do in terms of weight change or climbing speed, right? Because you would imagine that an animal that doubles the amount of running around it does with the rotation would get much better, show much greater weight change or change in climbing speed than an animal that only shows a 10% increase in activity. And so here we plotted the amount of activity change we see. So how much of an increase in activity do we get versus how much of a weight change we get in these animals. And we have the data for females on the left and the data for males on the right. And in the females, we see something that was unexpected for us. We basically see a positive relationship. So if you exercise more, you actually gain more weight. So the more exercise you do, the more weight you gain. And in the males, we don't see any relationship at all. There's actually potentially a reason for why you might see this. Females often have more body fat than males. And so if we're converting body fat to muscle mass, muscle is heavier than fat. So that might be one of the reasons. And that's one of the things we're looking into. But the data for climbing speed really isn't that great, that exciting either, because there's really no predictive value here for how much activity these animals will do, how much exercise they're doing, and how much their climbing speed changes. There's really no link here. There's no correlation at all, either in the females or in the males. So the amount of exercise they do is actually not a good predictor for this outcome in terms of climbing speed, which was surprising. So what these additional analyses showed us that the animal activity levels in our case are not linked to lifespan. We have a correlation between how much exercise they're doing and weight change in females, but it's going, well, in the wrong direction for what most people would like. And we see no link between the amount of exercise and the change in climbing speed. So what that really tells us is that the relationship between exercise and an exercise response is incredibly complex. And I can, and I'll tell you a little bit more about that in just a minute. So what I hope to have showed you today is that flies are a really efficient model to use for exercise studies because we can grow lots of them in a short period of time, and we can do very controlled studies. I hope I've also convinced you that, like humans, Drosophila vary in how they respond to exercise. So there's lots of variability. And hopefully you're convinced now that genotype and sex are important predictors for how an individual responds to exercise and how the exercise affects the individual. We have identified several genetic pathways, including some in the central nervous system, that are really important for these exercise responses. And we've unfortunately also confirmed that the relationship between the amount exercised and the exercise responses is incredibly complex. The things that we're really interested in now is to look at two candidate pathways controlling these activity levels. So one of them is the polycomb silencing system, which is a system that works on turning on and off genes. And so that's, I'm very excited about that. And another one that we're hoping to follow up on is the notch signaling system, which is a very conserved signaling pathway that is responsible for a variety of developmental processes, but also it determines when your stem cells are activated to, for example, repair muscle and things like that. So that's a really exciting one to us as well. one of the really big questions that we would like to get a handle on is how does exercise change behavior outside of the exercise session? So at this point, we've only really looked at how much do the animals exercise and then try to link that to the exercise response. But we really don't know what the animals do the rest of the day, right? If you think about yourself, if you've gone through some, heavy physical work or a really hard workout, you might actually go and not do a whole lot for the rest of the day, right? And so if that's the case, if that's a general tendency for animals to do, if you make them work out, that they then adjust their behavior the rest of the day, that actually means we might not really change their overall energy expenditure very much, right? If they adjust their activity downwards. Or if they, we make them exercise and then they're going to go eat extra, right? Again, then that would mean that we're really not expecting a weight change because we didn't actually shift that energy expenditure. So we really would like to learn more about what does an exercise treatment do to the behavior of the animals for the rest of the day? What are they doing outside of that 2 hours where we're watching them? And then obviously we would also like to understand the impact of different exercise treatments. Does it matter if you exercise in the morning or the afternoon? And the data for rodents really suggests that if you're exercising more in sync with your circadian rhythm, that might really be important. Unfortunately, the circadian rhythms also differ between individuals, so that gets very complicated. But so we really would like to understand what's the impact of an exercise treatment in a young animal, an older animal, in the morning, in the afternoon, how does that all connect to each other? And so those are the kind of questions that we're thinking about. And with that, let me acknowledge the people in my lab that did all this work. The people currently in the lab following up on this work are Bree Minifield and Heidi Johnson, and my tech, Katie Barden. But the majority, pretty much all of the work that I've shown you, the data was collected by my former PhD student, Louis Watanabe, with the help of a technician, Mina Momini, and 2 great undergrads, Michael and Cameron. And we've had the help of over 50 undergrads since 2013 working on this and other projects. I have 2 collaborators, Dr. Reed at UA, that was instrumental in developing the Treadwheel system, and then Tim Nagy here at UAB. And we had a little bit of funding for a pilot project that got us started on this project from NORC. And with that, I'm happy to take any questions. Thanks.
Thank you very much, Dr. Riddle. That was fascinating. I am looking in the chat and I don't see any questions. I don't know if you see any on your end.
I don't think so.
Well, I'll start us off. I've been reading about studies that have to do with gaining weight, humans gaining weight in response to exercise, and apparently it's pretty common, and it's due to something that you mentioned, altering behaviors, eating more, for example, because you're hungrier after having exercised. So how does that connect to this research? Is that not a purely behavioral response? Could there be a genetic component to that behavioral response?
So there's a lot of behavioral patterns that actually have genetic contributions, right? So there's definitely, so one of the things I've read a lot about recently is these, we talk about exercise, but the ecologists often talk about disturbance. What happens when you disturb an animal and it makes it move more, right? Those are the same kind of things that we're studying, only we're calling it exercise, they call it disturbance. And when they do disturbance studies, for example, with birds, there's definitely birds that are shy. And from the same species, you can have birds that actually don't mind if there's pedestrians walking by all the time and if there's, noises from traffic and stuff. So there's definitely behavioral patterns that are controlled by genetics. And so, they can actually show that the offspring of the birds that, don't mind the traffic noise, that they also don't mind. So there's clearly some genetics there. I don't know about food choices after exercise. I'm not aware of anybody having actually done genetic analyses of that. But, I wouldn't be surprised, that the response that you have is impacted by some of your genotype. But yeah, no, I mean, we would we currently don't have any data from the flies, at least on what happens in terms of feeding after exercise. But the data from humans, I think, is pretty clear that there's a lot of us that do tend to, go eat a bigger meal after the exercise.
Exactly. Okay. Thank you. Looks like Scott, I don't know how to pronounce your last name. Is it Brand?
Brandy, just like the drink, right?
All right, Dr. Brandy, please feel free to ask your questions.
So I just have two that I thought might be, you've probably already studied, but the levels of variation that you were studying are largely based upon the genetic line that you collected. I was wondering, since I'm probably one of the older faculty members here, whether there might be some research you've already done with regard to the different hierarchical levels of variation. In other words, the lowest levels for the individual. Mm-hmm. And so are you able to track individual flies by marking or some kind of other device through these days of exercise? That's number one. And if you were able to track individual flies, is there any research that suggests that the level of response for exercise varies with age? And even though this is a compressed sort of lifespan, if you were to be able to track individuals with regard to age, I would imagine that there are all sorts of behavioral changes that are ultimately controlled genetically that might be an input to the variation or maybe even the non-significance of differences that you're seeing as well.
Right, so we can definitely do these assays on just individual flies. That's possible. All of these studies that I've showed today are done on groups of 10 animals, just to get a little less noisy, I guess. But yeah, you can do these studies on individual animals and follow them throughout life. We haven't done any studies to follow them long term. So flies live, 60 to 90 days or so. So that's a lot of following them, but I mean, it's doable. We do know that with age, they do tend to move less. So they definitely move less as they get older. Even if you leave them alone. And the other thing that comes to mind is that there was a paper that was just published within the last 6 months, which was very interesting, where they looked at climbing speed of the animals as they got older. And what they could show that the climbing speed as they got older, and so basically their physical body condition or whatever you want to call it, was actually not related to lifespan. So it didn't matter if you weren't really good at climbing up the size of the tube anymore. That was not a good predictor for lifespan. So this like body condition in terms of how well you're able to run was not a good predictor for lifespan in these older animals either. So that was interesting.
There was one other thought that crossed my mind with regard to male behavior. Might not some of the exercise response differences ultimately be related to mate location and age of reproduction with regard to the differences in behavior?
That's totally possible. Yes, that it's a little funny in that we actually, I am not aware of anybody knowing actually if it's the males that move more to find mates in the wild, because we don't study fruit flies in the wild that much. Most of our studies are in the lab where they're in the same bottle, and so they don't have to run around very hard to find the girls. But I think that's a really good hypothesis, that is very likely that the males are just more used to actually being the ones that go and try to find the girls. Yeah.
Thank you.
Sure. Those are great questions. Thanks.
Thank you. Does anybody else have any questions you'd like to enter into the chat or say out loud? Feel free to jump in.
I'll jump in if that's okay.
Yes, please.
Hi, Nicole. Great presentation. I actually really enjoyed it. I'm curious if, I don't know if you did this and I missed it, or if anything else is known about it, but the loci that are associated with an increase in activity when you trigger it, when you stimulate them, is that the same? Is there anything known about overlap in those loci and then spontaneous activity when it's not actually stimulated?
So we've compared them, and the overlap is very little. So there's really very little overlap. And we're not necessarily sure why. But I mean, I think part of that has to do with the fact that for them to respond to the stimulus, obviously they have to be able to sense the rotation. You know, there has to be all kinds of signaling happening between the sensory organs that know that the rotation's happening, and then the brain, and then back to the muscle to actually make stuff happen, right? So we do tend to see, for any of the response type measures, we tend to see signaling show up more than we did before. So that definitely seems to be more important for the responses than for baseline. And so, you know, there isn't a ton of overlap. There's, you know, usually we don't have a lot of genes that overlap. It's usually like, you know, maybe a handful.
Anybody else? What about diet? And some of the early charts that you showed, that there was a big jump in the obesity rate in about the 80s. I don't know if something changed in our diet. And then the map that you showed of the states with the highest obesity rates, I mean, a lot of them were southern states, a lot of upper Midwest. You've got a lot of carbohydrates, a lot of fat, a lot of sugar in the diet. To what extent does your study tell us something about that?
So I have to admit, purposefully, I've been staying away from diet because that's what Dr. Reed at Tuscaloosa studies. She focuses very much on diet. And so I try to not step on her toes. But the diet definitely has a big impact on how you will respond to an exercise treatment. And so her work, she uses high fat and low fat diets and also high and low sugar diets to try to then understand how that changes the genetics, right? Because there's going to be different genes that are responsible and different pathways that are going to be activated by these different diets and how that interacts with the exercise. So there's basically a diet by exercise interaction effects that you can totally see. And so that's why I think it's going to be incredibly difficult to really design exercise treatments for a person, right? Because the optimal exercise regime will not just depend on your sex and your genotype, but it'll probably also depend on what diet you're currently on. And your optimal exercise regime probably will have to change when your diet changes or when you get older. And so it's actually a really complex problem. And right now, and I think that's part of why so many people get frustrated, right? We put them on an exercise treatment that we know works for a good amount of people, right? And then it doesn't work for them and they're incredibly frustrated, and then they just give up rather than just realizing it wasn't their fault. We just hadn't found the right one that's matching what they need. You know, and so, yeah, I wish the answers were a little simpler, but unfortunately they're not.
And presumably the Drosophila's diet is constant throughout the testing, regardless of activity level.
Yeah, I mean, they're basically on ad lib food, but it's the same food throughout. So they live on a cornmeal, sugar stuff.
but the female fruit flies, for example, that have gained more weight with an increase in exercise, their diet has remained constant. Is that right?
Yes. Yeah. I mean, for those females, then that's something we have to work on a bit more is, so in general, females have a lot more body fat. And if we just think that a lot of reason why humans also tend to hit these plateaus or gain weight when they exercise is that if you convert fat mass to muscle mass, you actually increase weight because muscle mass is denser and heavier.
Exactly.
And so I think that's one of the things we need to rule out to figure out if these animals actually, I mean, they must have just gotten denser because, I mean, insects have exoskeletons, so they can't get bigger, right? So they have to get denser. And so we think that that's probably a conversion of fat to muscle mass. And Dr. Nagy's core facility we should be able to sort that out with his help.
That is fascinating. Thank you. Who else has a question for Dr. Riddle?
Those are all really good questions. Thanks, everybody.
So are we looking at in the future for the impact on human exercise, the collaboration of a physical trainer, a nutritionist, many different players in order to achieve optimal results for humans
I mean, obviously,
exercising?
some of the people that do these kind of studies in humans from the Center of Exercise Medicine might be able to answer that better. But yeah, I think that's what it looks like. We are really going to have to. We need basically a lot more information about a person before we can actually find the right exercise treatment for them. I mean, I think right now it is often just a trial and error thing to just, you try different things and see if it works. And it's great for the people where it works. It's not so great if you're the ones that are these, what they call non-responders, or the ones that have the terrible opposite response, right? Yeah, so.
Okay, well, you've given us a lot to think about a very relevant and important topic, fascinating research. I want to make sure that no one else has any additional questions. Well, thank you again. Let's thank Dr. Nicole Riddle once again for her excellent work.
Well, thanks everybody for giving me the opportunity to share my work. I appreciate it. And if anybody has any questions, feel free to email me. My lab's always happy to happy to talk.
Thank you so much.
Thank you.
Bye, everyone.
Bye, everyone.